So this is question number 2. Now let's move on to the question. Okay. The treatment of patients with antibiotic can affect the composition of their gut flora. Gut flora are important as they help protect the body from pathogens. State the meaning of the term pathogen. So what does the term pathogen means? It means that it means an organism which can causes diseases. Pathogen is an organism which can causes diseases. See the answer. A pathogen is an organism that causes diseases. Okay. And state the meaning of the term antibiotic. What does antibiotic means? Antibiotic is any kind of drug or medicine that directly kills or destroys any kind of microorganism or bacteria which is growing in your body. Okay, so now let's see the answer. An antibiotic is a chemical substance or drug medicine that directly kills or destroys or inhibits the growth of bacteria. Okay, bacteria or microorganisms or pathogen. So now let's move on to the next question. The effect of an antibiotic on the proportion of different types of bacteria A, B, C, and C, D, and E in gut flora was investigated. So it's an investigation where the effect of antibiotics on the proportion of different types of bacteria was seen. So step 1 is the type of bacteria in the gut flora of a group of people were determined. Step 2 is half the group was then given a dose of antibiotic and the other half was given a control. And step 3 was the types of bacteria in the gut flora were again determined. The diagram shows the proportions of the types of bacteria in the gut flora. So this is the diagram which shows us the proportion of the bacteria before being given the antibiotic, after being given the control, and after being given the antibiotic. Okay, so we can see different types of trends over here, like the amount when before giving any antibiotic there was a huge number of bacteria type C compared to A and E, but when the control is given, the number of A increases, C decreases, but E remained constant. After giving the antibiotic, we can see a change that the proportion of D increased. C became really low, like it decreases up to 50 to 70%. And we can see that B is in a really small amount. So now let's go with the question. State one conclusion about the effect of the control on the variety of bacteria in the gut flora. So we need to give one of our conclusion. What happens after giving a control solution to the bacteria? So we can see through the diagram that the level of C decreased, A increased, and E remained constant. So we can just give that answer. So let's say the answer: increases A, decreases C, and has no effect on the proportion of E. Now let's move on to the next question. Estimate the decrease in the proportion of bacteria type C in the group given the antibiotic. So we need to estimate. We don't need to calculate. We just need to estimate, like guessing what decrease has happened in the proportion of C after giving the antibiotic. So we can see through the diagram it has decreased up to 70 to 80%, right? So we can estimate it by 70% or 80%. That would be enough. Like we can give it 70% decreased. It's enough. one more question this way now. So now let's move on to the next question. Explain how treatment with antibiotics can affect the gut flora. So when we treat through the antibiotics cannot distinguish between the good and bad bacteria. So it will try to kill or eliminate all the bacteria present in our gut flora, including the good and bad bacteria both. So if we take it, and if we are in prescriptions with antibiotics, then we need to keep in mind that they do not discriminate. That they do not discriminate, and they will kill both the good and bad bacteria in our gut flora. And that's why we can see that both types are inhibited, they cannot grow properly, and antibiotics affect different types of gut flora bacteria in varying ways, and it leads to changes in the proportion of bacterial population and due to the death or inhibition. Okay, so let's see the answer. Okay, so we can see that treatment with antibiotics can affect the gut flora because antibiotics do not discriminate between beneficial gut flora and harmful pathogenic bacteria. As a result, both types can be destroyed or inhibited. Additionally, antibiotics affect different types of gut bacteria in varying ways, leading to changes in the proportions of bacterial population due to the death or inhibition of some species and resulting competition for space and nutrition among the remaining bacteria. Okay, so I guess this is the end of question 2. See you in the next question.